Namaste students, Hamasha Khushro. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, Women's University, Jaipur. So today we are here to discuss the topic pH, pK and buffers. So these topics comes under the subject fundamentals of biological chemistry. So let's start. So what is buffer? Buffer solutions are the aqueous solutions consisting of mixture of a weak acid and its conjugate base, or a weak base and its conjugate acid. So an important property of buffer solution is its ability to maintain a relative constant pH value in response to addition of small amount of acid and base. So what a buffer does, that if you add in a solution some acid or some base, then if buffer is already present there, it will resist to change the pH of the solution. Okay. So additionally, the pH of the buffer solutions remains relatively stable also during dilution. For this reason, buffer solutions are used in a wide range of chemical applications, primarily as reagents that allow to maintain a constant pH. Okay, so the pH, pK and Henderson equation, Hazel-Belch equation. Okay, so the pH is, what is the pK and the pH? pK is the pH value at which the chemical species will accept or donate a proton. Okay, so pK is a specific pH value. So the lower the pKa, the stronger the acid and the greater the ability to donate proton in the aqueous solution. Okay, so pK and pH, however, it is only an approximation and should not be used for concentrated solutions or for extremely low pH acids or high pH bases. So once you have pH or pK values, you know certain things about the solution and how it compares with other solutions. So the lower the pH, the higher the concentration of hydrogen ions. Okay, so the lower the pKa, the stronger the acid and the greater its ability to donate the protons. So basically pH depends on the concentration of the solution. This is important because it means a weak acid could actually have a lower pH than a diluted strong acid. Okay. For example, concentrated vinegar, acetic acid, which is a weak acid, could have a lower pH than a diluted solution of hydrochloric acid, which is a strong acid. So on the other hand, the pK value is constant for each type of molecule. It is unaffected by the concentration. So pK is a constant value. Even a chemical ordinarily consider a base can have a pKa value because the terms acid and bases simply refer to whether a species will give up protons or remove them. Okay, so for example, if you have a base Y with a pK of 13, it will accept protons and form YH, but when the pH exceeds then 13, YH will be deprotonated and become Y. So because Y removes protons at a pH greater than the pH of neutral water, that is 7, it is considered as a base. So these were the basics of the pH, pK and the buffers. So hope you are satisfied with this detailed session. If you have any query, please write in the comment box. I will resolve your query. And this session is powered by digital version 2 Jyoti Vidhyapeeth Women's University Jaipur. Thank you.